Now we're going to continue looking at some other germicidal categories, but we'll go through these quickly. The main ones I want you to recognize are the alcohols, hydrogen peroxide, uh, chlorine, detergents, and soaps. And so halogens are used in germicidal preparations. They are chlorine, which it denatures proteins by disrupting the disulfide bonds. It's an intermediate level germicidal preparation. It's unstable in sunlight, inactivated by organic matter, but we use it in water, sewage, wastewater, and on inanimate objects. Iodine interfused with disulfide bonds, proteins. It's an intermediate level germicidal. It's milder medicine and dental degerming agents. It's also used as disinfectants and ointments. So here they show the application of some of the halogen compounds, chlorine gas, that's used in large-scale disinfection of drinking water, sewage, and wastewater. You have the hydrochlorides, which are bleach, used extensively in sanitation and disinfection of food equipment. You notice that common household bleach is a 5% solution of sodium hypochlorite. And so dilutions of 1 to 10 to 1 to 1,000 are highly effective as germicides. Okay. All right. And so here you see the cantaloupes are disinfected with chlorine treated water, kills salmonella on their surfaces. A lot of vegetables and fruits, they use chlorine to clean them. Iodine. This is a patient be treated with povidone iodine solution in preparation for surgery. Okay, this is phenol, which is a carbolic acid. Is a poisonous compound, disrupts cell walls and membranes and precipitate proteins. Is low to intermediate level germicide. So use it as a bactericidal, fungicidal, virucidal. So it's not sporicidal. So it does not kill spores, but will kill vegetative states of those. You'll see this in Lysol and also in antibacterial additives in soaps. All right, so like I said, we're just going to go through some of these kind of quickly. Okay, alcohols. Only ethyl and isopropyl alcohol are suitable for microbial control. So they're intermediate level, and the concentrations of 50% and higher dissolve membrane lipids and coagulate proteins of vegetative bacteria and fungi. Water is needed for proteins to coagulate. So thus alcohol shows a greater microbicidal activity at 70% concentration than at 100% concentration, which means in order to coagulate protein, you need some water because it needs to undergo hydrolysis. And so you need water for hydrolysis. So if you have greater than 70%, the effectiveness of alcohol diminishes because you don't have water to come in there and break things apart. So here's some alcohol at a nurse's station. Nurse takes advantage of an alcohol gel hand cleansing station. They're very common in hospitals and clinics to encourage hand antisepsis. Then you have hydrogen peroxide. It produces highly reactive hydroxyl-free radicals that damage protein and DNA while also decomposing to O2 gas that makes it toxic area for anaerobes. So antiseptic at low concentrations. So strong solutions are sporicidal. So hydrogen peroxide is what contact lenses use. So disinfection of contact lenses with peroxide-based cleaner. The bubbles are produced, indicating the release of oxygen gas. That's important to know as well for the quiz at least, but it's kind of good to know if someone that has contacts and look at the solutions on your uh, cleaners for the contact lens. You'll see hydrogen peroxide, ethyl alcohol, and then solutions of 70 to 95% are germicidal. It's very inexpensive. It's non-irritating. It's uh, limited by its rate of evaporation. So primary applications use skin degerming agent and antiseptic. Surfactant action removes skin oil, soil, and some microbes sheltered in deeper skin layers. Occasionally used to disinfect electrodes and face masks. Then you have rubbing alcohol, which is isopropyl alcohol. It's used in some disinfection of objects, surfaces, and limited skin cleansing. So it's more no, microbicidal and less expensive than ethanol, but these benefits must be weighed against its toxicity. Inhaling its vapors can adversely affect the nervous system. So if you have too much and breathing in too much isopropyl alcohol could be dangerous. You have, once again, the hydrogen peroxide. 3% hydrogen peroxide is most common form, useful in treating infections by anaerobic bacteria because of lethal effects of the oxygen released. And then alkalides, you have glutaraldehyde, which is a 2% solution, very high level, kills very well. Used as a sterilant for heat and sensitive uh, for heat sensitive instruments. And you have formaldehyde, intermediate to high level. It's a disinfectant, preservative, and toxicity limits its use. And then this is a technician treating surgical instruments with glutaraldehyde prior to heat sterilization. So a lot of times we use multiple modes of sterilization. Gases, gases and aerosols. Example, prepackaged milk supply sterilized by ethylene oxide sterilizer. Glutaraldehyde is a sterilant that's used for materials that are damaged by heat. So like respiratory therapy equipment, hemostats, fiber optic endoscopes use glutaraldehyde. And these are some of the other ones, but detergents and soaps. Detergents are polar molecules, and so they're used on surfaces. And you have what they call the quaternary ammonia compound. They access surfactants that alter membrane permeability of some bacteria and fungi. It's very low level. Soaps, once again, mechanically remove soil and grease containing microbes. Weak microbicides destroy only highly sensitive forms. So detergents and soaps are effective in actually getting the microbials off of our skin and off of areas that you're trying to clean. They're very effective at getting off grease and oil that the microorganism can stick to or in, so it stays on surfaces, stays on your skin. Use detergents and soap to get them off of your skin, off of the area of concern, inanimate object, and being able to rinse and flush them. Here's detergent and soap is saying there's millions of bacteria in a number of days. If you have a soap that is a non-germicidal soap, so you have quite a bit of over a period of days when you clean with non-germicidal soap. If you don't, uh, you clean but don't rinse, then you still have a high level of microbials present. If you have a germicidal that's in the soap as well, then you see the number of bacteria decreases over time. Okay, so you can look at these. And it's very important in terms of washing. And which of the following kills by producing oxygen radicals? Yep, you knew it. hydrogen peroxide. Then heavy metals. So solutions of silver and mercury can kill vegetative cells in low concentrations by inactivating proteins. Once again, think about, because you see this a lot, what's a vegetative cell? That means vegetative means it has undergoing metabolism and binary fission or reproduction. So it's active. That's vegetative. Heavy metals are low level killers, but they do have some response. This is a petri dish with a lot of bacteria, and you have a little bit piece of gold, you have a little piece of silver amalgam. That sounds familiar, doesn't it? Silver amalgam is what you use in, right, fillings. And so just the nature of the metal makes it where you see that around the silver, you have limited bacterial growth. Around the gold, you have limited bacterial growth. So that's why we use it in fillings. And then here's some of the heavy metals and what they're used for. And the dyes actually have a low level antimicrobial effect, and they're very active against gram positive species of bacteria and fungi. Okay, so sometimes dyes are used in antisepsis and wound treatment. And then you have acids and alkalis, low level activity, and you look at the different acids that are used. And so here's some of the active ingredients of some of the common things we use: Lysol, Tilex, Ijex, Lysol disinfecting spray. And you can look and see the different things that how the chemical agents and the activity, the antimicrobial category. Purell hand sanitizer, Pine Sol. I'm not requiring that you know all these names, but it's just interesting seeing uh, what they're used for. Okay, let's get started with chapter 12. Okay, so now we're going to start with chapter 12, and we'll go through it fairly quickly. Scoping out the chapter. So, discovery. Antimicrobial drugs come from many sources. Most of them, called antibiotics, are produced by certain bacteria or fungi. Others are synthesized through chemical reactions alone, and some are made by combining those two methods. So we take a do no harm, and so selective toxicity. So antibiotic literally means against life, but the actual intention of these drugs is to target only microbial life. This is a very important guiding principle that drugs do no harm, do not harm humans while they are getting rid of the infectious agent. And then we're going to look at how antimicrobial drugs stop infections. So drugs are chemicals that can interfere with some specific microbial structure or function, such as the cell wall, cell membrane, proteins, DNA, RNA, ribosomes, or metabolic pathways. So while in contact with the drug, the microbes are either destroyed or severely inhibited and can no longer grow. So then we look at toxicity and other side effects. Promising antimicrobial therapy is spoiled by the numerous possibilities of adverse side effects. Drugs can harm body tissues and organs, disrupt the normal microbiota that help keep a balance in the body's organs, and induce allergies and hypersensitivities. Then we have drug resistance. It's a race. Microbes are very adept at rapidly altering their physiology and genetics to adapt to drugs, making them less effective. They may also develop enzyme systems that dismantle the drugs. They may block the drug's entrance, expel the drugs, or use an alternate pathway that bypasses the drug's effect on them. Then we have to choose appropriate drugs. So selection of an effective drug is guided by several factors. Among the most important considerations are the nature of the infectious agent, knowing which drug it is sensitive to, the possible side effects of the drug, and the medical condition of the patient. So here's some of the principles of antimicrobial therapy. The goal of antimicrobial chemotherapy is to administer a drug to an infected person that destroys the infective agent without harming the host cells. So antimicrobial drugs are produced naturally or synthetically. The perfect drug does not exist, but by balancing drug characteristics against one another, a satisfactory compromise can usually be achieved. So here's some of the characteristics of the ideal antimicrobial drug. Selectively toxic to the microbe, but not toxic to the host cell. Microbicidal rather than microbistatic. Microbicidal is it kills the microbe, and microbistatic means that it slows it down. We want it to kill rather than slow it down. If you just slow it down, that means that after the course of the antibiotic, it will come back, or it may come back. Another ideal characteristic is remains potent, remains strong enough, and strong long enough to act, and is not broken down or excreted prematurely. Another ideal characteristic is not subject to development of antimicrobial resistance. So it needs to be able to kill them without the microbes learning how to uh, be resistant to it. Another thing is that it complements or assists activities of the host defenses. So these are important for you to know, the characteristics of the ideal antimicrobial drug. So the ideal antimicrobial drug also remains active even when diluted in body fluids and tissues, that it's readily delivered to the site of infection. It can get to where the infection is, that it's reasonably priced, and that it does not disrupt the host health by causing allergies or predisposing the host to other infections. So antibiotics are common metabolic products of aerobic bacteria and fungi. So antibiotics, common metabolic products of aerobic bacteria and fungi. So the molds in genera penicillin and cephalosporium, that tells you which antibiotics were created from those molds. Penicillium, actually, that we get penicillin from there. Cephalosporium, we actually get cephalosporin. Find the the origins of the antimicrobial drugs. They inhibit other microbes in the same habitat. So the antibiotic producers have less competition for nutrients in space. They actually secrete what we now call antibiotics so that they have less competition for nutrients in space. Excuse me. So we have origins of antimicrobial drugs. You see here, there's different areas that if you have mold here, let's say, then it's going to cause the bacteria around it not to be able to grow. And so selected microbial sources of antibiotics. Penicillium, this comes from a mold, and you get the penicillins. Cephalosporin is a mold, get the cephalosporins. Micromospora is from bacteria, you get gentamicin. And the bacillus, you get bacitracin, it's from bacteria, bacitracin. And so the streptomyces is a filamentous bacteria, so it's bacteria as well. But from streptomyces, you get a number of very common antibiotics. Okay, here's a terminology chart that is very important for you to know. Prophylaxis, antimicrobial chemotherapy, antimicrobials, antibiotics, semi-synthetic drugs, synthetic drugs, narrow spectrum, broad spectrum. It's important to know these. So this is a slide that's important to know. So we're going to actually talk about them. Prophylaxis, to keep guard before, so a process that prevents infection or disease in a person at risk. Chemotherapy is use of drugs to treat disease. Antibiotic is against life. More specifically, antibiotics are against microbial life. So antimicrobial drugs should be selectively toxic. They should only kill or inhibit microbial cells without damaging host tissues. So this is an important point. As the characteristics of the infectious agent become more similar to the vertebrate's host cell, complete selective toxicity becomes more difficult to achieve and more side effects are seen. So that's why it's fairly easy to get antimicrobial agents for bacteria, because bacteria are prokaryotic and we as vertebrae are eukaryotic cells. And so we're so different that if there's something that will uh, affect and become toxic for a bacterial cell, it's easier for us not to be affected by it. But the closer that the parasite or the infectious agent is to our cells, the harder it is to find antibiotics and antimicrobials that will kill it without affecting us. So 5 major components that are useful drug targets in an actively dividing cell. So one of them is inhibit cell wall synthesis, break down the cell membrane, interferes with function of DNA and RNA, inhibition of protein synthesis, and blockage of key metabolic pathways. Here's some of the actions that microbial groups, the actions of the drugs on the different microbial groups. So you see the penicillins, they have cell wall damage and lysis. So encephalosporin, cell wall, bacitracin, cell wall damage and lysis. So you notice in a lot of ointments, they put bacitracin in there. Typically, we don't have any problem at all with bacitracin. That's because it disrupts the cell wall and has damage to a cell wall, but we don't have cell walls. So all these are pretty effective in killing the microbe without harming us. Then you start going with the others. Inhibit ribosomal protein synthesis. Erythromycin does in cell wall for vancomycin, but you look at the different ways that these common antibiotics affect the cells. Okay, and this is more of the, in terms of fungi, loss of membrane permeability with different inhibits DNA, RNA synthesis. So these are some of the ones that are effective, but you see the closer they are to us, the more it could actually affect us. And see, with viruses, we do have some antimicrobials. And so you see with some of these, how it prevents viral budding, stop viral replication, blocks formation of DNA and RNA strand. These are some of this is common one, AZT. If you heard ATT, that's this one, blocks formation of DNA from the RNA strand. So that's one I was telling you about in class, where it blocks the reverse transcriptase. Okay, here's some of the major targets so we could see more schematically. So this is a bacterial cell. So you have cell wall inhibitors. This is a cell wall. So these affect the cell wall inhibitors. Then you have cell membrane, cause loss of selective permeability. And so that's this blue part. This is the part that's affected. Then you have DNA and RNA that it inhibits the replication and transcription. So it inhibits the gyrase and inhibits polymerase. So these are the different ones that will affect the DNA and the RNA. And then you have some that affect the protein synthesis inhibitors acting on the ribosomes though. And so there's some that act on the site of action of the 50S, site of action of the 30S, and both the 50 and 30S. This is where it affects. This is the 50S portion of the ribosome for protein synthesis, and this is the 30S. So these are different drugs that will act on the ribosome. Then you can have metabolic pathways and products that are blocked. This is some of the sulfa drugs, trimethoprim. And so what happens is you start with a substrate, you have an enzyme that will produce a product. And these drugs actually block that metabolic process, blocks the enzyme. So then we have different spectrums. So that's the range of activity of a drug. Narrow spectrum, they're effective on a small range of microbes. Okay, so target a specific cell component found only in certain microbes. Then you have a medium or broad spectrum, greatest range of activity, target cell components common to most pathogens. And this just shows some of the drugs that are available and whether they have broad, intermediate, narrow, and also a narrow on the other side, but still narrow. So typically we look at gram positive, gram negative. So you look at these drugs, tetracyclines, quinolones, chloramphenicol, they have a very broad range. They affect gram positive, gram negative, rickettsia, chlamydia, and mycoplasma. They do it all. Then you have intermediate, and so they have gram positive, gram negative. That's usually the biggie. Does not affect rickettsia. And then there's some effect on mycoplasma and chlamydias. Then you have narrow group. This is where we have the penicillins. Even though penicillin was the first antibiotic, what we found out later is it was only effective on gram-positive bacteria. But gram-positive bacteria make up a lot of bacteria that affect us. So it was very effective, but these are limited to, these drugs, antibiotics, are limited to gram-positive. And some of these can actually do some gram negative as well. Then you have narrow group 2, which are these, which actually affect the gram negatives, but they don't affect the gram positives. And they don't affect the rickettsia, chlamydia, and mycoplasma. You don't have to know these. It's just kind of good to know in general that you have. Well, you do have to know that we have broad spectrum, intermediate spectrum, narrow, which is gram positive only, and narrow as well, which is gram negative only. You don't have to know the names though. So a drug that targets lipose polysaccharides in a gram negative cell would be considered what? That's selectively toxic. So they will go into specific drugs that affect the bacterial cell wall. I'm not going to require that you know these specifically, so we'll go through this quickly. And it talks about affecting the cell wall, it damages the cell wall. And this is with the membranes. You see the drug comes in, gets on the cell membrane, and literally breaks apart the cell membrane. Okay, I'm not going to require that you know that specifically. Drugs that affect nucleic acid synthesis, so block the synthesis of nucleotides, inhibit replication, or stop transcription. So we have some antiviral drugs that are analogs, so they look like some of the purines and pyrimidines, and so they actually insert themselves in the viral nucleic acid, prevents replication. And drugs that block protein synthesis. Okay, so ribosomes in eukaryotes differ in size and structure from prokaryotes. So antimicrobics usually have a selective action against prokaryotes. They can also damage the eukaryotic mitochondria, so we have to be careful about that. Because eukaryotic mitochondria is thought to have been previously a prokaryote that was engulfed by a eukaryote. And so if you have a substance that will be effective against the prokaryotic bacteria, we may actually have some damage to the mitochondria in different cells. So that's something we have to be careful about. And this is some of the targets we have. Like you can have a drug that will come in and will affect the 30S. Will block the messenger RNA from reading uh, the codes on the transfer RNA. This is another one where another antibiotic will actually block the peptide bonds from forming when you have amino acids trying to join. This is another one where another drug will actually block the anticodon site on the tRNA, so it cannot combine with the messenger RNA, blocking protein synthesis. In tetracyclines, it blocks the other site of the tRNA, so this tRNA can't come in and join up the amino acids to the next tRNA. Erythromycin blocks the messenger RNA here, so it cannot read. The tRNA cannot read this messenger RNA. Once again, proteins are not produced. So then you have drugs that affect metabolic pathways. So metabolic pathways, you have competitive inhibition. So the drug competes with normal substrate for the enzyme's active site. So that means that the drug that's sent in, it actually, the substrate that the enzyme will try to actually work on cannot go to the active site and actually have its activity. So synergistic effect, the effects of the combination of antibiotics are greater than the sum of the effects of the individual antibiotics. So that's just saying that if you have two antibiotics, some of them work together and actually have stronger effect than each one individually. So that's why we have multi-drug treatment in some situations where the two of them together or three of them together work much better than individually. And so this is competitive inhibition, where you have metabolic pathway needed to synthesize THFA, contains two enzymes that are chemotherapeutic agents, targets. Under normal circumstances, the PABA acts as a substrate for the enzyme, and the product of which is needed for eventual production of folic acid. So this is let's say a bacterial cell. It needs folic enzyme in order to synthesize purines, pyrimidines, and amino acids. Okay, so it needs folic acid. In order to get the folic acid, it takes PABA and creates the dihydroteric acid and hydrofolic acid, and hydrofolic acid needs to go to folic acid. Well, this drug stops this process to go from dihydrofolic to folic acid. Once you stop this, then you won't get the folic acid. So these sulfa drugs stop the PABA from going to hydroteric acid. So once we stop this, then none of this will occur, and you can't get folic acid. So ultimately, you do not get purines or pyrimidines or amino acids. So what's important about this is only certain bacteria form this step. And so only certain bacteria affect this step. So that means that if you use sulfa drugs, will not affect humans in terms of stopping this process to get folic acid, just the bacteria that actually rely on this step to get folic acid. And that's part of competitive inhibition. Here's an example. Here's PABA, and here's the enzyme that enhances this step to actually take PABA to the next level. It steps in here, it reacts, and then you get the next step. Well, what happens, the sulfa drug kind of looks like the PABA, but if you have enough sulfa drug in here, the sulfa drug gets in and keeps the PABA from getting into the enzyme, and then this enzyme is left where it cannot do its job, and so you cannot produce folic acid. Okay. Okay, so survey of the major antimicrobial drug groups. And so the drug groups, like I said, I'm not going to require you to know the different drug groups, but just know that antibacterial drugs that act on cell wall, know these major themes that you have some that act on cell wall, antibiotics that damage bacterial cell membranes, well, act on the bacterial cell membranes, the drugs that act on DNA or RNA, drugs that interfere with protein synthesis, and drugs that block metabolic pathways. Okay, let's go over that again. So what I want you to know are these parts that are highlighted or bolded. You have antimicrobial drugs that act on cell wall. You have some that damage bacterial cell membranes. You have some that act on DNA or RNA, and you have some that interfere with protein synthesis, and then some drugs that block metabolic pathways. And so I'm not going to go into which ones affect what. So I'm going to zip through some of these. This is subgroups and uses of penicillins. So you can go through it if you want, but I'm not going to require these of you. Cephalosporins. Okay. And they have different, with cephalosporins, they have different generations. And each generation gets more and more coverage. It gets better, less toxic effects. So you have different generations of different drugs. Okay. So, like for example, with cephalosporins, the first generation, it was most effective against gram-positive cocci and a few gram-negative. But as we modified it, as we give different generations, you get down to the fourth generation, which cefepime is a cephalosporin. It has the widest range. So it has both gram-negative and gram-positive coverage. Okay. I'm not going to ask you specifics for these. That's not going to zipping through them. Sulfa drugs. There's different newly developed classes of antibiotics. It's good to kind of just look at them and you can decide if you want to know more about them. But you look at, here's some new drug types. They're formulated from pre-existing drug classes and 3 new drug types. And yeah, you can look at the different ones and what they affect. Okay. And then we have antifungals. The cells of fungi are eukaryotic, which present special problems in chemotherapy. The great majority of chemotherapeutic drugs are designed to act on bacteria, generally ineffective in combating fungal infections. Drug that is toxic to fungal cells is also capable of harming human cells because they're eukaryotic as well. So there are 5 main groups of drugs that have been developed to specifically treat fungal infections. And I'm not going to have you specifically know these, but you can look at some of the things that you've seen before, like amphotericin B, nystatin. They bind to fungal membranes and cause loss of selective permeability. So you see some of these, risofulvin, a stubborn case of dermaphyte index, but it's nephrotoxic. And so synthetic azoles, ketoconazole, miconazole, you've heard of these different things. They're broad spectrum. And so we do have a few, but like it was saying, they're eukaryotic cells as well. So we a lot of times have a lot of toxicity associated with them. Okay, this is, you see Flagyl, antiprotozoan drugs, and that's antiparasitic. So antiviral chemotherapeutic drugs. This is selective toxicity is almost impossible due to obligate intracellular parasitic nature of viruses. That's just a simple way of saying they use our body's own cells to actually manufacture their cells. So if we chew something that kills them, it will actually kill us. And so we have to get very, very selective in terms of what we do to kill them to make sure it does not harm ourselves, our own selves. So this is the things that we focus on. We try to focus on blocking the penetration into the whole cell. So for this is for virus, this is antiviral. So we try to block penetration into the whole cell. Then we try to look at blocking replication, transcription, or translation of the genetic material. And these are some of the antivirals that we have currently that will do that. And then we try to prevent them from maturing, so maturation of viral particles. We have protease inhibitors in HIV, and that's another way that we try to fight them. Then we have drugs for treating influenza. Remember, influenza is typically from viruses. So these are kind of interesting. We'll, I don't know if you heard of amantadine and rimetadine. At one point, they were looked at as they were like the saviors for flu. But look at look at this. Essentially, we had to discontinue them as flu treatments. Why? Because current viral strains have developed almost a complete resistance to them. We use them so much, and for everyone, and just everyone that comes in, give them rimetadine or amantadine, and it killed off the ones that were susceptible. But that means that the flu viruses that were left were ones that were not susceptible to these. And so essentially discontinued it because just about all viral strains now are resistant to them. So we have what they call Relenza and Tamiflu. So they're slightly broader spectrum. It blocks neuraminidase and influenza A and B. So these are some of the drugs that inhibit virus entry or release. This anti-HIV drug, once again, I'm not going to ask you specifically, but sometimes it's kind of interesting in terms of reading up on some of this. So this one, Maraviroc, covers up the cell receptors. So a cell that when HIV comes to a cell, it attaches to a receptor. This covers up that receptor. Then you have the Tamiflu and Zamifir drugs do not stop the early influenza cycle involving entry, so it lets the flu virus get into the cell. It doesn't stop the uncoating or the replication. What it does after a virus cells gets in and takes over the metabolic system, then this particular drug, Tamiflu, blocks the action of neuraminidase that is required for final budding and release of new viruses. So whichever cell is infected, that cell is a goner, but it prevents the budding or release of new viruses. And here's a schematic. And it's just saying with some of the drugs that have this HIV and it blocks the receptors. So this HIV virus cannot get into the cell. This is another one that blocks the receptor, and so it doesn't even attach. This one it attaches, it's just it can't get through. This one it doesn't even attach; it just floats around in the serum. It does not get into the cell. This particular one lets the influenza virus get in, so it attaches to the receptor, it gets in, it takes over the DNA and the ribosomes, and then it produces more virus. But see, the virus, as it gets up to the cell membrane, it buds off or pinches off, and it takes part of that cell membrane, and it will now make more new viruses. But with this particular drug, it prevents the budding. So that's this one here that we talked about, this Tamiflu. It blocks the action of neuraminidase as required for final budding release of new viruses. So that's where it prevents. So this virus cannot leave. Okay. Then you have anti-herpes, acyclovir, and then oral and topical treatments for oral and genital herpes, chickenpox, and shingles. And so in inhibition of nucleic acid synthesis, this is the this is the reverse transcriptase inhibitors. So stop the action of reverse transcriptase in HIV and blocks DNA synthesis. All right. And so, like I said, I'm not going to ask you these specifically. I think it's just kind of interesting for you to see them and know. Okay. This is more in terms of the HIV drugs, their action, HIV drugs. So it's good to go through and kind of know what's out there. And once again, I'm not going to ask you to know these. Now we have drug resistance. Drug resistance is an adaptive response in which microorganisms begin to tolerate an amount of drug that would ordinarily be inhibitory. So result in genetic versatility and adaptability of microbial populations. It can be intrinsic, which means it has it in its own makeup, and it can be acquired. It learns it from something else. So the acquisition of resistance is the main problem for microbial chemotherapy. This is drug resistance. Acquisition of this drug resistance is the main problem for microbial chemotherapy. I mean, you think about it. We find different drugs that will kill microbes. As they learn how to offset the antimicrobials, sometimes we almost look at it like it's a losing battle. Sometimes they learn, seems like, as fast as we get new antibiotics. And that's very distressing. And we're working very hard on that. Let me tell you let's look at some of the reasons that they're able to learn so quickly. So how does resistance develop? It's newly acquired after spontaneous mutations in critical chromosome genes. So sometimes you have mutations. That's where a lot of people think, oh, that's where most of the resistance comes from. That is not where most of it comes from, but it can occur. Then you can have acquisition of new genes or sets of genes via transfer from another species. That's big. So they transfer from another bacteria. So they teach each other how to be resistant. So here's the different ways that you can have intermicrobial transfer. So it transfers resistant factors. They're little pieces of RNA or DNA encoded with that drug resistance. So the transposons duplicated and inserted from one plasmid to another or from a plasmid to the chromosome. Okay, let's look at that. So let's say here's a bacteria that is dead. So it dies, but let's say it had a, a DNA fragment that had a resistance gene in it. Let's say this DNA fragment had a resistance gene in it. So even though this is dead bacteria, this one comes around that's alive. This fragment is just floating around out here. It gets incorporated into this bacterium. Now this bacterium now learns what this one knew about resistance. So this resistance gene gets incorporated into the gene for this bacteria. This bacteria now has learned the resistance, whatever that resistance was. That's from a dead bacteria. Here's another way it can get transferred. This is a bacteria that has resistance gene. This one doesn't. And so it can transfer part of the plasmid that has the resistance gene in it. And when it transfers the plasmid, now that plasmid gets incorporated into the DNA or RNA of this new bacteria, and it teaches it about the resistance. And actually, this is another interesting way. So this is transformation when it just absorbs the DNA from a dead bacteria. This is conjugation where bacteria come together and they communicate and it has transfer of information. This one's kind of interesting. You know, viruses can infect bacteria. So if you have viruses in a bacteria that has resistance, it can actually incorporate that DNA or RNA into itself and infect another bacterium. When it infects this bacterium, it transfers the resistance gene so that this bacterium will now learn and have resistance. That's a virus that can transfer it over. And that's called transduction. Very interesting. Okay, so here's some of the mechanisms of acquired drug resistance. Sometimes the bacteria can learn how to inactivate the drug. So inactivation of a drug like penicillin or by pilocinase, it's an enzyme that the bacteria produces that will inactivate penicillin. So then you have decreased permeability. Sometimes a bacteria will, this is a normal cell receptors, and the drug comes in and it gets into the cell. Well, sometimes bacterium actually learn how to reject that drug coming into its cell. Okay. Then we have activation drug pumps. Sometimes a drug can get into the cell, but if it's inactive, then the drug gets into the cell and does whatever effect it's going to do. But if they have an active drug pump, drug gets in and the bacteria pumps it right back out. And so you just have drug that does not do its job inside to kill the bacteria. And also they can change the binding site. So remember when we said that some drugs will come in and stop replication and stop transcription translation. And so it will keep the messenger RNA from attaching to the transfer RNA. And so some bacteria will find a way to, instead of having this site that this can connect to, it has it where it changes the site that the drug would affect. And so now this drug runs around and it does not have anywhere to dock to stop this process. So it literally just changes the site where the drug would go. And so that's another resistance. And then alternate use of alternate metabolic pathway. Let's say if a drug will affect this pathway, so the drug stops this from happening in order to get the product inside of bacteria. Well, some bacteria have found a way when a drug comes and the sequence metabolic pathway is blocked. Some bacteria have found a way to bypass that step and go to other intermediates to produce the product. So now you have drug that's there that stops folic acid, let's say, production, stops that. Well, the bacteria just found a way to go around it and produce the product a different route. And so that's using alternate metabolic pathways. Okay. And so this is which of the following effects do antiviral drugs not have? So antiviral drugs at this point do not have ability to kill the extracellular viruses. So viruses outside of the cell, they found a way to stop viruses from duplicating, stop viruses from attaching, but we at this point do not have a way to kill the viruses. Okay. So large populations of microbes likely to include drug-resistant cells are due to prior mutations or transfer plasmids, so no growth advantage until exposed to the drug. So then if you expose, the sensitive cells are inhibited or destroyed while the resistant cells survive and proliferate. So eventually the entire population will be resistant. That's natural selection. Let me show you a picture of that. Let's say there's In a population of microorganisms, here's a population that are um, not resistant to whatever drug we have out there. So all these microorganisms, let's say they're bacteria, they're not resistant. So which means we give them a certain drug, and so they are all killed off. But there's a few in the population, there's a couple that are resistant. But so someone gets better, when it kills off all these bacteria. But when these bacteria are gone, so these bacteria are dying with the antibiotic, then these two start producing more because now there's not as much competition. So they start producing more. And so if you continue exposing these bacteria to drugs that these are sensitive to, then they continue killing these off. And so when there's fewer and fewer of these, this is the one that's sensitive to the drug, then the ones that are resistant will take over. And so they become very prevalent. And so eventually you get to the place where all you have left are resistant bacteria. So microbial cells involved in an infection, most sensitive cells are eliminated by a drug, then most new cells are resistant. That's just describing what we just went through. So strategies to limit the drug resistance of microorganisms. One is drug usage. Physicians have the responsibility for making an accurate diagnosis and prescribing the correct drug therapy. You have the correct drug therapy, you give the drug that will kill all of them that are out there. You don't just, so you kill all the ones that are out there in that particular individual. So you have to have compliance with the physician's guidelines. Once the physician has a responsibility of accurate diagnosing and prescribing, then you follow the physician's guideline. So the patient should take the correct doses by the best route, appropriate period. This diminishes the selection for mutants and can resist low drug levels and ensures elimination of the pathogen. So combined therapy with two or more drugs together increases the chances that at least one of the drugs will be effective and that a strain resistant to one drug will be eliminated by the other. So sometimes what we have to do is give more to accomplish what we need. So the resistant to one, if we have a second drug that kills that one that's resistant, then you do not have a proliferation of the resistant. So we need to do more drug research, focus on developing shorter term, higher dose antimicrobials that are more effective, less expensive, fewer side effects. And so we're continuing to search those long-term strategies. Proposals reduce the abuse of antibiotics, range from educational programs for medical workers to requiring justification for prescribing certain types of antibiotics. A lot of physicians just give antibiotics willy-nilly, but once you give more antibiotics to more people, more bacteria are exposed to it, kills off the ones that are sensitive, but then leaves around the ones that are resistant. So more resistant ones start showing up in the ER. So sometimes we have over um, prescribing, and especially valuable antimicrobials may be restricted in their use to only one or two types of infections. What this is saying is that when you have certain antimicrobials that are very broad and they're very specific and kill certain things very well, don't use them unless you specifically have that organism. And then when you use them for that organism, the, then you combine them with other things to kill other organisms that may be resistant. That way, you don't have it out there enough where those organisms can find resistance. Because the reason being that that's so important is if the bacteria learn about some of our big guns and they become resistant to them, we may not have antimicrobials that can fight off the resistant ones. So we limit the use. So then we can have the addition of antimicrobials to animal feeds must be curtailed. Because currently, a lot of times in animal feeds, they will give animals some of the antibiotics that we use so that they can limit how much infection the animals have. Well, when the animals have it, then it gets rid of the um, bacteria that are sensitive to that, but it enables the bacteria to that are resistant to proliferate and teach other bacteria about the resistance. So if we continue to, and this is done a lot worldwide, where our antibiotics are given to animals, and before we're even able to get the benefit of them, the bacteria show up that are resistant very quickly because they've learned about it from animals. So government programs that make effective therapy available to low-income populations should be increased. We need to effectively treat low-income populations, not just the rich people get good antibiotics and kill off, and then low income, they just kind of fend their way, do the best they can. It will generate more resistance. And then vaccines should be used whenever possible to provide alternative protection. And then now we talk about interactions between the drugs and the host. 5% of all people taking antimicrobials will experience a serious adverse reaction. That's some of the side effects. Some major side effects: direct damage to tissue due to toxicity, allergic reactions, disruption of the normal balance of normal flora. Then you can possibly have super infections. Here's some of the interactions. This is the one that I showed you in the book, where it talks about tetracycline, where it could cause discoloration of the teeth and gum, the teeth, if you had taken it when you were young. There's a super infection where you get an infection, and so you give a drug to treat the infection. Well, it kills off all the bacteria. So in the intestines, it didn't even target those. It wasn't targeting the intestines, but in the intestines, it kills off the good bacteria. This is normal microbiota, important to maintaining intestinal health. But in our intestines, you have some that are pathogens. Well, when you kill off all these good, then these few that are pathogens, they start proliferating. So you get what you call a superinfection. These are pathogen overgrowths. So after you have give an antibiotic to take care of strep throat, then before you know it, you have an intestinal disorder, that you have overgrowth of pathogens that were resistant to that antibiotic, and you can get what they call a superinfection. And then some of the adverse reactions, penicillin, ampicillin, what you can actually get. Some of them cause diarrhea, enterocolitis, and these are some of the side effects of some antibiotics currently. Adverse drug reactions, once again, you don't have to know these, but these are some of the ones that are common. So anemia, immunosuppression from AZT. Okay, so when you're selecting antimicrobial drugs, you need consideration must be given to at least three factors. So the nature of the microorganism causing infection. So specimens should be taken before antimicrobials are initiated. So you take samples before you even give uh, antimicrobials. And then the degree of the microorganism susceptibility, how susceptible they are. So you determine the minimum inhibitory concentration. That's the smallest concentration of a drug in a series that visibly inhibits microbial growth. And then you look at the overall medical condition of the patient. So think of those three: the nature of the microorganism causing the infection, the degree of the microorganism susceptibility or sensitivity to various drugs, and the overall medical condition of the patient. And here's some of the different disc diffusion tests. This is like if you had different antibiotics. So this whole petri dish is just full of bacteria. And so you put in different antibiotics, these different sites, they're all the same size, and you measure to see how much of the bacteria around each of these this antibiotic kills. So on this one, this particular bacteria was pretty sensitive to this one. Okay, it wasn't very sensitive to this one. So you wouldn't use this antibiotic for this particular infection. You would use this one, or even this one. And so, and here you can see what they're trying to show here is that this is antibiotic. This is antibiotic. You see, this has a pretty good range. Goes around like this. Pretty good range of coverage. This one has pretty good range of coverage. But if you notice something here, this one typically would stop around here. You can see how the range is. This one would typically stop around here. So what's interesting feature is these bacteria in between the two that normally wouldn't be affected is affected because because of this one with this one close by, the interaction between these two has a better kill rate than each one individually. So if you put these two together, you actually kill more than if they were individual. Okay. And this is another interesting, you see this one has gentamicin, which is GM, and as trionam, which is this one. So when you're trying to decide what's the better antibiotic to use for this particular bacteria, you look and you say, well, they both have, about the same range of kill. They both have the same range. But if you notice in this one, there's little clusters. You see these little clusters that are in the periphery that are still colonies. These are colonies of resistant bacteria. So there's still some bacteria that is in this culture that will be resistant to the gentamicin, even though most of them are killed. So this is definitely one of choice because otherwise you kill off the main ones that are sensitive to it, but now you're going to leave behind all these others that are resistant, and these are going to start taking over. Then you have a resistant strain that is causing infection. This is just another diffusion test. What it is you actually put different um, amounts of antibiotic. It goes down to almost no antibiotic here and gets a higher concentration as you go up. So the higher concentration goes up, higher concentration goes up. And then you set this down in the petri dish and you see it kills up this way really well. And then as it gets down further, as there's less and less antibiotic, then it gets to the place where it doesn't have hardly any kill rate. And there, at that level, and the best concentration to actually kill this particular organism. Same thing with this one. This is at this level, and this concentration is the best kill rate. You look and see what the best kill rate is. And this is two dilution tests, and this is the minimal inhibitory test. This says that you have control. So this is just the microorganism, and you have a little bit of antibiotic, a little bit more, a little bit more, a little bit more, a little bit more. And you see the amounts of antibiotic you have, and you get to a certain point where it's clear. So at this point, if you have this much antibiotic, then uh, it effectively kills the microorganism. And this is just another example of that as well. You can read that if you want. Okay. So in a two dilution test, the minimum inhibitory concentration is the what of a drug that is required to inhibit growth of microbe. But what is it? In a two dilution test, the minimum inhibitory concentration is the blank of a drug that is required to inhibit growth of microbe. Is it the largest concentration of a drug? Is it the standard dose? Is it the smallest concentration or is it the lowest dilution? Right? It's the smallest concentration. Is it the smallest concentration of a drug required to inhibit growth of microbes? All right, you guys do well. Um, I, I I don't know if I have to look at my time to see if I went over, but um, we will have the quiz on this and on Wednesday. And I just wanted to emphasize to you all, let's see, make sure that you know the definition of microbiological contaminants, know the definitions of like that chart that has disinfection, pasteurization, chlorination, filtration, all those different, know the different filtration methods, and specifically HEPA filters. Um, know the strength, resistance of different microorganisms, like easiest microbial forms to kill, hardest to kill, and then I don't ask you specific names. I did ask about the uh, what you use in soft contact lenses. What do you use uh, to use in soft contact lens disinfectant? Know about food irradiation and how it's utilized. Once again, know the definition of like degermination and antisepsis and sterilization and disinfection, all those different definitions. Know the difference between bacteriostatic and bactericidal. And know what happens with microorganisms when you actually freeze them. Do they die? Do they slow down? What happens with that? Know thermal death time, what that actually means and how it works. Know what the definition of antibiotics are. Know what narrow spectrum drugs, broad spectrum drugs, semi-synthetic, synthetic drugs. Know those different definitions. Know the characteristics of antimicrobial drugs. They readily delivered to the site of infection, highly toxic. Toxic, not just slowing down, do not cause serious side effects. Remember all those. Know how superinfections can occur, and know that why there are fewer antifungals and fewer viral drugs as opposed to bacterial. Know the reasoning. Look at drug susceptibility testing. That's what we just went over. And then look at the different factors that influence a physician's decision to prescribe different antimicrobials. And then also look at allergic reactions to drugs and know the minimal inhibitory concentration, what that is, and know what it means in terms of when you say, you know, uh, mismanagement of antibiotics, antibiotic abuse, allergies, prophylaxis. Know what prophylaxis is. Uh, and then also, once again, think of the different things that can be taken into account when deciding on which drugs to actually use. Okay, I just went through all of um, I just looked at the quiz and described all the things that you need to know or be associated with for the quiz. So go through it again. Make sure you read the chapter. And you should do fine. Okay, look forward to seeing you on Wednesday. Have a good day.